Today we are going to discuss the different proteins involved in replication process. In the first part, already we discussed what is replication, what is the mode of replication, what is origin point of replication in DNA. Okay, now today we are going to discuss different types of proteins required for this replication process in prokaryotes. There are several types. We'll discuss some important proteins here. One of the important protein is DNA A protein. DNA A protein, it will bind with that specific region of the origin point. Okay, that means it initiate DNA replication process. Also open up the DNA duplex at origin point. First, it will open up where? At the origin point. That is the main functions of this DNA A protein. Now, come to another important protein, that is DNA B protein. This is also called as helicase. Helicase is the enzyme which can break the hydrogen bonds, because already we know DNA are double-stranded molecule. These two double strands are joined with the help of hydrogen bonds. To separate this double-stranded DNA molecule, hydrogen bonds will be break. So this DNA B protein or helicase that is involved in that process. That means to unwind the DNA duplex. Unwind the DNA duplex. Okay. Another important protein, that is DNA C protein. Actually, DNA C protein is required by DNA B protein, so that this DNA B protein can bind with the specific segment of the origin point. Okay? DNA C proteins are required by DNA B proteins, so that DNA B protein can bind with that specific region of the origin and they can unwind the DNA duplex. So rather we can say DNA C as a loading factor, for DNA B. Okay, another important point, that is another enzyme, that is gyrase. Okay, it shows the activity of topoisomerase type 2. So what is the function of this gyrase or this topoisomerase? As we know, this DNA are double helix structure. These are bind with the help of this hydrogen bonds. Suppose these two double helix are joined with the help of hydrogen bonds. Now, what is the function of this DNA B or helicase? It will break down these hydrogen bonds and unwind the DNA. That means it will open up. Okay, in that way. So when it will open up from this point and this helicase not completely open up all the hydrogen bonds. Okay, like this side already they are double stranded and this side open up due to breakdown of this hydrogen bonds. So when these two strands are separated, it creates tension in that region. So it starts to supercoil like this. Supercoiling occurs. It will create tension when this DNA strand gets separated like this. The Y-shaped structure, it will get separated. So here it will create tensions. So supercoiling occurs. To relax that supercoiling or to relax tension, this gyrase enzyme cut the DNA strands to relax supercoiling. So there are actually two types of topoisomerase are there, topoisomerase 1 and topoisomerase 2. Topoisomerase can cut only single-stranded DNA and relax this supercoiling, and topoisomerase 2 can cut both strands and relax that supercoiling. So what is the function of this gyrase? To relax the supercoiling or to relax the tension. Okay, that is the function of this gyrase. Another one important point that is called as single stranded binding protein. In short, it is called as SSB, single stranded binding protein. What is the meaning of that? Because see, there is a double helical structure, hydrogen bonds are there. Suppose in that position hydrogen bonds breakdown occurs. How it will break? Due to this helicase enzyme. Now it is open up. Due to breakdown of hydrogen bonds. And as complementary nucleotides are there, they again try to produce these hydrogen bonds and become double-stranded. So this single-stranded binding protein will bind here. So what is the function? It will prevent to form again the double-stranded DNA. Okay, that's why function is same, single-stranded binding protein. The name itself shows its function. That means this protein will bind with the single strand of the DNA double helix. So why? Why do bind? Because by binding with this single strand, it will prevent to form again the double helix. Okay, that is the function of this single stranded binding protein. Another one important protein that is called as primase. Primase. What is the function of primase? To synthesize primer. To synthesize primer. Regarding this enzyme, we'll discuss more detail in the mechanism part. Right now, here you just know that primase is the enzyme which is responsible for the synthesis of primer. And primers are nothing but some short nucleotides. Some short nucleotide sequences. Okay, what is primer? Why primer required? We'll discuss in the mechanism part. Okay, another one. Enzyme, important enzyme, that is phosphorylase. Okay, phosphorylase. So what is the function? It aids phosphate group. It aids phosphate group. That enzyme also, regarding that function also, that mechanism also we'll discuss during the replication mechanism part. Okay. So up to here we are discussing total 7 proteins. Okay. First one is DNA protein. So it will bind at the origin point, and at the origin point it open up the DNA duplex. Okay. Another one, that is DNA B protein. It is also called as helicase. It will bind with that origin point. Origin point means the point where the replication starts. Okay. So here it will break the hydrogen bonds and unwind the DNA duplex. Another one, that is DNA C protein, that we can say is a loading factor for DNA B. Another one, that is the gyrase. What is the function of gyrase? It shows the function as topoisomerase. So it will relax supercoiling. Okay. Another one, that is single stranded binding protein. So when this helicase enzyme break the hydrogen bonds, the DNA double helix becomes single-stranded. But remember, at once all the DNA double helix not become single-stranded. Okay, the process of DNA replication continuous, and this hydrogen bond breaking also is a continuous process. Okay, now this is a primase. The function of primase is to synthesize the primer. Primer means some short nucleotides. Okay, another one, that is phosphorylase. What is the function of phosphorylase? It aids the phosphate groups. So regarding these two enzymes, we will discuss later on in the mechanism part. Okay. Now we are going to discuss another two important proteins, that means enzymes, which take part in the replication process. Another one important is, this is ligase. Ligase can join the DNA fragments. Okay, to join the DNA fragments. To join DNA fragments. Now it says is ligate. Okay, joins the DNA fragments. Another one, the enzyme, which is involved in the whole process, that is called as DNA Polymerase. That is called as DNA Polymerase, because this polymerization means the increasing the number of DNA. That main role that is played by this enzyme, DNA Polymerase. Okay, and in case of prokaryotes, there are three DNA polymerase are there. DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. There are total three types of DNA polymerase in case of prokaryotes. In case of eukaryotes, there are several types of DNA polymerase are there, like alpha DNA polymerase, beta, gamma, epsilon, delta. There are different types of in case of eukaryotes. But now we are discussing in prokaryotes replication process. So these three DNA polymerase involved in the replication process. Okay. First, this DNA polymerase 1, it has exonuclease activity. So what is exonuclease activity? What is nuclease? Nuclease, ASE. Normally when it is end with ASE, that it's denote an enzyme. Okay, so nuclease is an enzyme which can break these nucleic acids. Okay, so nuclease can also be two types. One is called exonuclease. Another one is called as endonuclease. Exonuclease means it will break this phosphodiester bond from one end, maybe in the 5 days end or maybe in the 3 days end, one after another it will break. And endo, it means in the middle part. Middle part means it's not like that it will only break here only middle part. Okay, but in case of exo, suppose it is 5 days to 3 days and one after another it will be nucleotide will be breakdowns like that. That is called exo. Okay, there are two types of nuclease. Now here only we are discussing about exonuclease. So two types of, again, exonuclease may be removed from 5 days to 3 days time also, or 3 to 5 also. Okay, this DNA polymerase, this has both 5 days to 3, also this 3 days to 5 days exonuclease activity. What is this? This is exonuclease activity. This DNA polymerase has both 5 to 3 exonuclease activity also, 3 to 5 exonuclease activity also. Okay, and main function of this DNA polymerase 1, its function is to DNA repair and gap filling. That is the main function of this DNA polymerase 1, DNA repair and gap filling, and it has both 5 to 3 and 3 to 5 exonuclease activity. But in case of DNA polymerase 2, it has only this 3 to 5 exonuclease activity. 3 to 5 exonuclease activity, and it also take part in DNA repair mechanism. And lastly, the DNA polymerase III, it also have 3 to 5 exonuclease activity. Okay, and DNA polymerase III is the main replicating enzyme. It is the main replicating enzyme. Also, it has proofreading like DNA repair, like it has also the proofreading activities. Proofreading activity. What is the meaning of proofreading? Like, have you heard that proofreaders, suppose in a newspaper is a proofreader, suppose in a book is a proofreader. Proofreaders means he will see this actually is all correct or not. Okay, like that. In case of DNA polymerase also, it will aid the nucleotides. How it aid nucleotides? In a semi-conservative way. That means based on this old strand, the nucleotides which are present in that strand, suppose here adenine is there, then it will aid thymine. So here guanine, here cytosine. But sometimes some error may occur. Okay, sometimes unknowingly some errors may occur. So it will find if some wrong nucleotides are joined. Okay, if some wrong nucleotides are joined, that will be removed. How remove? With the help of this 3 to 5 exonuclease activity. And again, it will correct that one. That one is called as proofreading activity. Okay, so up to here we discuss about the function of DNA polymerase. DNA polymerase is the important enzyme which is take part in polymerization reaction in DNA synthesis. In prokaryotes, there are 3 types of DNA polymerase. DNA polymerase 1, it mainly take part in repair and gap filling. Another one, DNA polymerase 2, it will take part in repair part. And DNA polymerase 3 is the main replicating enzyme which is take part in replication process as well as in proofreading activities. Okay, proofreading means it will correct the errors during polymerization process. If some wrong nucleotides are added, then it will try to remove that one and give the correct nucleotides. That is called proofreading activities. That proofreading activity is very important process. Okay, this is the function of this DNA polymerase. Now, we'll discuss this DNA polymerase III. This DNA polymerase III, it has several subunits. There are several types subunits are there. But here we'll discuss some important subunits like alpha subunit, beta subunit, and this epsilon subunit. Subunit, it will play in the polymerization reaction. Okay, another beta subunit, it will act as a clamp. What is clamped? Clamp means it will give the optimal processivity. So this is double stranded DNA. When hydrogen bond will break down, it will be open up. So this polymerase will bind here, DNA polymerase, for polymerization reaction. So it will act as a clamp, so it will not be fall down easily from this strand. Okay, to give optimal processivity. Another one, epsilon subunit, it gives the proofreading activity. Already we discussed this proofreading activity. That means it will give the 3 to 5 days exonuclease activity. So these are the some subunits of DNA polymerase III. So up to here we are discussing about the proteins which are take part in the replication process. In my next classes we will discuss the mechanism of replication. Thank you.